All right, welcome back. Gene regulation in prokaryotes. Right? Bacteria, cells that don't have a nucleus. See, everything's different because if you have a nucleus, then the genes have to be regulated by other molecules. And of course, they're stuck in the nucleus, so there's issues with getting through the nuclear membrane. You'll recall that in transcription, the messenger RNA has to eventually leave the nuclear envelope, as it's sometimes called, and get outside into the endoplasmic reticulum. So with prokaryotes, they don't have the nuclear membrane or an organized nucleus, so for them it's more of just a big soup. So things are slightly different. So for gene regulation, we've noticed some key differences, but this one is a good one to start with because it's a more simpler sort of process. And essentially, what we mean by gene regulation is that you have to be able to turn genes on and off because genes make proteins, right? That's what they do. They code for proteins. They don't actually make them, but we've seen the process. So genes make proteins, but organisms need to regulate how much of those proteins they have. So you have to be able to turn genes on and off. When the proteins they need get low, you need to be able to turn on the gene and crank out some more. But if you have lots of them, then you need to be able to say, hold on, gene, stop making protein. We don't need it. So they have available. And so not only can genes replicate themselves and make proteins, they can also regulate themselves. There are sort of little DNA sequences that don't make a protein. Instead, what they do is they regulate how the other part of the gene that makes the protein is expressed. When we talk about a gene being expressed, we say a gene, when it makes its protein, it's being expressed. That's what that means. It's expressing itself. It's making itself known to the world. Look, I make melanin. Ha-ha, and it's making melanin. Genes are expressed. So available. All right. So what we're going to do is we're going to use an example. The example we're going to use is the regulation of a gene that actually makes enzymes for the digestion of lactose. Okay, so that's going to be the example. It's the one your textbook uses, so we'll use the same one. And I just have to pause for. Okay, so let's go back. I'll erase this dot, and let's look at how the gene is set up. So we're going to draw a picture. Okay, so here in black, I'm going to draw a sort of a sequence of DNA. I'm going to draw it straight rather than curly, and I'm going to draw it as a bar. So this is not the two strands of DNA. This is just basically looking at, well, I guess it could be the two strands of DNA. What we're going to find is we're going to put a little purple box right here. This represents a section of the gene, which I'll color in with a nice purple crayon. Like that. Then we're going to get a green box a little further down the line, maybe right about here. Okay, and we'll color that in with a nice green crayon. Yep. Then we'll get a little sort of, I don't know, orangey section. I'm just going to use the same colors as your book so that everything looks similar. So here's an orangey section. Sort of right beside the promoter. And we'll color that in with an orange crayon. Nice, nice. Orange. Like that. Good. Then over here, we're going to have three sections. They're going to be sort of different shades of red because these are all the coding parts of the gene. So let's start with red. We can leave a little gap there. Then let's do a lighter red. Let's see if we can get like a, oh, where are my colors? Maybe a pink. Pink. Okay. Pink. Better do the crayon on that too while I've got it. Oh, that's the wrong one. Pink. There we go. And red. And these three are all sort of touching each other. And then one other color. Let me see what I can find. Something you can see. Something that's sort of reddish. Well, maybe this darker pink will work. Let's see what it looks like. Okay. Oh, pen. Pen. Dark pink. Kind of the same pink, isn't it? All right, let's pick this darker red then. That works. There. Color it in, crayon. All right, so these are very specific segments of the gene. Okay, I'm going to pause and just let you catch up with your color. Okay, so we should mention, I mentioned this and I didn't write it down, so I'm going to write it underneath as a title. Normally we put titles on top, but I didn't leave enough room. So I'm going to write underneath what this is. And this is the, we'll call it lactose metabolism. Right? Gene regulation. So we'll put this at the bottom. Okay, so to metabolize lactose, you will need an enzyme. What do you think that enzyme would be called? Lactase. And so the last three sections in the reddy pinky colors, these are three different regions of the genes that code for different enzymes for the digestion of lactose. So they make, if you code them, if you transcribe them and translate them as a protein, they make enzymes. So we're going to call them different names. So the red one is going to be called LAC-Z. LAC for lactase. The pinky one is going to be called LAC-Y. Can you see that? And the last one in the orangish color, or the reddy orange color, is going to be called LAC-A. Why not Z? Where's X, you might say? Don't worry about why they're called what they're called, because we're not going to get into studying the actual enzymes themselves. So those are just three enzymes. So on your paper, you might put a little star and say, these are just the enzyme codes for making whatever we need to digest lactose. Okay? The green one, I'll go to that one next because it's an easy one. The green one, what starts off every gene? The what? A starting codon, which is part of a segment called the promoter. Good. So this is the promoter section. This is the section that basically initiates the copying of this gene. So if this gene, right, the promoter is where, if we're going to copy this gene and transcribe it into these enzymes, that's where the RNA polymerase would sort of attach and open up the molecule and start copying, right, to make the gene. Now this orange part is the new little piece. It's called the operator. So let's put the operator in there. And it's the part that is going to be the regulatory section. The operator. Okay? Now this whole entire thing from the promoter, including the operator, and all of the enzymes that are coded, is called the lac operon. So the word operon is used to describe the promoter part of the gene, the operator part, and actually, you know what I'm going to do? Did you already write that word? I should have written it on the top because we need to write things on the bottom. No, it'll get too crazy. Let's just white it out. Sorry. I should have left it on the top because I want to leave the bottom open because we're going to be drawing. Well, we may draw some other pictures, but okay, you know what? Leave it. Leave it. We'll draw another picture. Leave it like that. The more pictures we draw, the more we memorize it, right? So if we keep drawing the same picture and summarize, that's actually a good learning activity. So while we're lecturing, we're also learning because we're drawing these pictures over and over. Okay, so that's called the lac operon. Now, it's called the lac operon because this particular operon is all about making lactases to digest lactose. But there are many operons. Obviously, every gene works this way in the prokaryotes. And so there could be other genes here for other things. Whoops. The purple thing? What purple thing? This one? We're not there yet, but Jack, good question. So the purple thing, what is it? That's the part that we have to look at now. Okay. So this purple thing, we're going to call it lac-L. Ken, purple. Lac-L. All right. So this is the part of the gene that isn't. It's way upstream. It's not actually part of the operon itself. So that means that this will always be copied. It's always being copied. This thing. So it's gene, or not just being copied, but it's always being expressed. And what it does is it makes a molecule. It codes for. So it codes for a molecule. That's filling up in the cells and is always around. So there's lots of it. And this molecule is called a suppressor. Because as we're going to see, what happens is if there's lots of that molecule, it will bind with the operator and clog up the process and prevent the movement of the RNA polymerase to copy this gene. All right, that's sort of our next step. So we have this lac-L, which is a site that is going to make this repressor. So why don't we put down here lac-L is transcribed into a piece of messenger RNA, and it makes a repressor. So we're going to make this repressor a different shape. We're just going to call it's just a molecule. So why don't we just kind of give it a weird shape like that. Crayon, color it in. It's supposed to be purple. Color it in purple. And we'll call it the lac repressor. Okay. Now we have to just kind of figure out how it all works, now that we know all the pieces. Yeah, when it codes, when it is transcribed and translated into a protein, that's the molecule that gets made. Okay, and because it's upstream, you can imagine that the polymerase reading this gene is coming this way. So it's going to hit this lac-L every time, and it's going to express it. It's going to make the protein no matter what. But when it gets here to the promoter, when it gets past the promoter, and the operator is going to control whether or not it finishes the whole thing and does the lactases or not. Okay? All right. That's good. So I'm just kind of... yeah. Let's go down in it and let's start a whole new picture now. Are you guys caught up? Yes? No? Okay. So there's two situations that could happen. One is when we don't have enough lactose, and one is when we have lots of lactose. So the thing about the repressor, let's go back for a second. This lac repressor can bind to lactose. Okay? If the lac repressor binds to lactose, that'll change its shape. Remember, everything is shape. So let's first consider what happens when there's very little lactose in the cell. If there's very little lactose, you don't need to digest it. So you don't need to be expressing all these genes and making all these proteins. That's a waste, right? You want to turn this on when lactose is high. So here's what happens. Let's just redraw very quickly the entire thing. Okay. Now, I'm not going to draw the colors every time. That's going to just be... Should I? Are you going to be upset by that? You like the colors? Okay. So up here, we have the lac-L. And I'm going to write them in. And then over here, a little bit further down, we have the promoter. Right? And then we have the operator. And then we have our three sort of sections. So what I'll do is just shade them all in with gray so we can see them as opposed to okay, so here's the first guy, here's the promoter, here's the operator, and then here are the three genes. Okay, and I'll write their names in them. Without the colors, we won't remember what they are. So I'll write in what they are, right? So this is is going to make that repressor protein. This is the promoter. I'll just put prom in there. This is the operator. I'll put operator in there. And these are the different lactase enzyme genes, right? They were what, Y, Z, and A, right? Or Z first, I think. Y and A, right? Okay. So this section of the gene up here is making this protein. So what I'm going to do is I'm going to draw it in a very specific shape. It's got this shape, let's say. Now, if there's no lactose around, this shape, this suppressor, will move over here and it will bind on the operator site. Okay? So, in the absence of lactose, okay, another way to say that, maybe a better way to say that would be when lactose concentration is low. Right? So it's not necessarily the absence of it, it just means that it's fairly low. When the lactose concentration is low, this suppressor, and if you want, you can write in here suppressor. The suppressor molecule binds on the operator site of the operon. When it's bound there, it prevents the expression of anything further down the line. So if an RNA polymerase is trying to express this gene, is trying to transcribe it and translate it, it'll hit this operator with it's sort of clogged up now because this suppressor molecule has stuck on there, and it'll just stop. Question? Repressor or suppressor? I guess in your book they call it the repressor. Is that what I called it before? Okay. They both kind of mean the same thing, right? It's the same kind of word. But I'll just make you happy and write this as the repressor molecule. Repressor, suppressor, it doesn't matter. So repressor. Okay? All right. Okay, so here's what you have to understand. It doesn't jump to the operator. It's being made all the time because there's nothing here to stop the RNA polymerase from copying or from expressing this gene. So the entire cytoplasm of the bacteria is filled up with this molecule. It's everywhere. Okay? which means that if it's everywhere, it has a very high chance of bumping into the operator, doesn't it? If you dip this gene into a solution full of this molecule, that repressor molecule is going to keep bumping into the operator and clogging it up. That's what you have to think about it as. Okay, so it's not moving there as such. It's not being made and going directly there. It's just this gene is filling up the cytoplasm with this repressor, and when there's no lactose around, that repressor molecule is constantly bumping into the operator, binding it up, and stopping the production of these enzymes. Okay, all right. Now here's the cool thing: when lactose if this happens, of course, that means the cell would not be breaking down any lactose, right? Because it doesn't have the enzymes. These enzymes over here are not being made, which means that eventually lactose would start to build up in the cell because you're not breaking it down. So a little while later, there will be lots more lactose. So as the lactose builds up, here's what happens. Now we have the same picture. Well, let's write something here instead. Hang on. Let's write. Suppress the operator site. Okay, so we look at this prevents the transcription of the lac enzyme enzymes, or the lac enzyme codes, I guess we should say, because they're not enzymes, they're codes for the enzymes that are on. Codes. Okay. Those guys on the end. The result of this is that no lactose is being digested, or let's put less lactose is being digested. Never say no, never use absolutes in biology. I've learned that over the years. Less lactose is being metabolized. Instead of digested, let's say it's being metabolized. Let's learn to use these words so they become part of our vocabulary. Therefore, lactose begins to accumulate. The concentration of lactose will rise slowly. Let's put that in brackets. The concentration goes up. As the concentration of lactose goes up, something interesting happens. So finish copying that. All right. So as the lactose begins to go up in concentration, here's what happens. So now we'll draw the next set of pictures. All right. So here's our little section of DNA again. Here's our first little dude. Here is the promoter and the operator, and then the three guys, and I'll quickly shade those in again in crayon. These two, and then the enzyme codes. Okay, so remember what these are. Just very quickly, we'll write them in. We have the promoter, the operator, I'll just put oper, and then we'll put the lac genes here. Doesn't matter which ones they are. Okay, so now I'm going to have another molecule building up here. I'm going to put this little black molecule. What this is lactose building up in the cell. It's building up because that repressor was stopping the production of lactase, and so the lactose is building up. Let's shade the lactoses in so they don't feel left out. Maybe a nice blue color. Somehow I think lactose would be blue. Here's an interesting science question. What color would molecules be if you could go on the magic school bus and shrink down and find a molecule? What color would they be? We know that sulfur is a yellowy substance. I wonder if a single sulfur atom would be yellow. There's a good research question for your thesis sum here. Okay, so now here's what happens. The Suppressor molecule is still being built, right? I'm going to go back to see what shape I gave it. Okay, there it is, the repressor. You want me to call it the repressor? That's fine. The repressor is being made. So let's draw a couple of these repressor molecules, right? Here they are. Here they are. La, la, la. Lots of them too, because they are constantly being expressed. There's no regulatory expression. But with all this lactose building up, what's happening is the lactose molecules are binding with these repressor molecules. And once they're bound here, their shape renders them now inactive. So they cannot bind to the operator site. Yes? Is that allosteric regulation? I suppose it is kind of a form of that. Yeah. Usually we use that term when we're talking about regulating a particular enzyme itself, not the gene that codes for it. But it is the same principle, isn't it? It's the idea that the binding of this molecule, when it binds the suppressor or the repressor molecule, it renders it inactive. In other words, it can no longer bind with the operator. And if it can't bind with the operator, then an RNA polymerase is free to carry on down the gene and express the lac enzymes. So let's write that underneath. Yeah, the lactose is attached. And remember, in biology, if anything attaches, it changes your shape, essentially, right? Because of the electrons all pushing and pulling. So this renders it inactive. So let's write a little statement to that effect. The lactose binds with the repressor molecule. Now, don't confuse it. It doesn't bind on the DNA strand. It doesn't bind with the code. It binds with the actual molecule that's floating around and prevents it from binding to the operator. The lactose binds with the repressor molecule, preventing it from attaching to the operator site. This allows RNA polymerase to continue along the operon, let's say. I'll put it back in a second. To continue along the operon, which is the whole sort of section, and express. By the word express, we mean transcribe and eventually translate the lac enzymes or enzyme codes. So the gene is now on. Continue along. Yeah, the lactose binds with repressor molecules, preventing them. I guess it should be plural or singular, one or the other, but I kind of went both there. So let's change it to grammatically correct, preventing them from attaching to the operator site. This allows RNA polymerase. So in other words, the operator is not clogged, right? Then everything can flow according to plan. Got what? Go down a little bit? Sure thing. I'll just pause and let you cut that. So in this case, the presence, or we could say high concentrations of lactose bind with the repressor to inhibit or to stop or prevent the expression of lactase. Okay? So in other words, we get a case where if the lactase goes up, if the lactose concentration goes up, the gene production goes up. Gene expression. You see that? Now, the opposite is also possible. It's possible that the binding with a molecule could make instead of inactivating the suppressor or the repressor, it activates the repressor. So this would be a slightly different sort of system. So this can work the other way. Gene expression goes up. The gene expression means if lactose goes up, then the lactose is clogging the repressor molecules, and this gene expression is allowed to happen. Gene expression means these three enzymes will be made to digest the lactose. And that will in turn cause the lactose to drop, which means that then the repressor will bind and shut it off. So it's responding to the increase in lactose. The increase in lactose is what turns on the gene. Lactose. The lactose is the sugar. The lactase are the enzymes that the gene makes to break it down, right? So that says lactose. As lactose goes up, the gene expression for lactase goes up, and then that helps to bring the... So it's like trying to... Remember I talked to you about equilibrium, keeping everything in balance? That's what's happening here, right? Yes, Lord? All right, this can work in the opposite way. Instead of disabling the suppressor, repressor, sorry. It's a mental block I have. I must have learned it that way back when. The repressor and enabling the gene expression, the operon expression, a molecule can attach to the repressor and activate it. Change its shape so that it can bind. So that would mean that when the concentration of something goes up, that would, well, it would mean the opposite. So let's think about this for a second. So this time, the molecule of lactose is attaching to the suppressor and deactivating it, so the gene is expressed, right? It's possible, though, that in the opposite case, when the lactose builds up, or when another molecule builds up, and it attaches, then it would attach to the operon after it's attached to the other molecule, which would shut off the gene. So this would mean that instead of raising concentrations of a molecule turning on the gene, rising concentrations of the molecule would shut off the gene. So it works in the opposite way, right? So rising concentrations of this molecule would shut off the gene. Expression. A molecule can attach to the repressor and activate it. So the next sentence is, rising concentrations of this molecule would shut off the gene expression rather than turn it on. So it just works in the opposite way. It means that in the first case, a bound repressor molecule does not latch on to the operator, and the gene goes. In this case, a bound repressor molecule would then latch on to the operator and shut off the genes rather than the opposite. So it's just the opposite idea, right? In your textbook, there's another picture, and they show a different, I think it's tryptophan expression, and it works in this opposite way. But we're not going to need to draw all the pictures. We just need to know it works both ways. That's all. Yep. Yeah. So the RNA polymerase, the question is, in the textbook, the picture has the RNA polymerase binding right here, in this picture, right? Actually, no, it's in the first picture, isn't it? Yeah, it's in this one. So the RNA polymerase, of course, remember, is the molecule that opens up this DNA. This is DNA, remember? And it begins to transcribe messenger RNA to be sending it to the ribosomes for the making of these three enzymes. So, yeah, the polymerase complex. Remember how in our last video we talked about how that moves down and expresses the genes. So that's what it would be doing in this case. So in your textbook, yeah, that's what its job is to express the gene, to make it into protein. And so the essential thing here is that when the repressor binds to the operator, it prevents the RNA polymerase from binding and doing its job. So none of this happens. But if you have lots of lactose and those repressors are all used up, binded up, and can't attach, the RNA polymerase is free to go. In the opposite situation, like with the tryptophan example, then we would just have the opposite happening. Okay, so I'm just going to conclude by saying have a look on page 334 in your textbook if you want to see the pictures that go with this opposite thing, and it's all about tryptophan operon. And you can see how the pictures would be slightly different. Okay, and we'll stop there.